Hi everyone, welcome to your online lecture for Anatomy and Physiology 2. In today's lecture we're covering digestion and absorption, and by the time you're watching this lecture my hope is that you have a fairly decent understanding of the anatomy of our digestive tract as we now get into the physiology. The primary function of the digestive system is to make essential nutrients available to our body for use. So to take what we consume, break it down to a point where we can absorb it, and then our body, our cells, our tissues can use these nutrients for energy. Our digestive system is quite comprehensive and involves 7 primary mechanisms. The first one is ingestion. This is the process of just taking food into the mouth, and this is where really it starts its way, starting on its journey through the digestive tract. Digestion is a group of processes that break complex nutrients that you consume into smaller ones. And really what that does is it facilitates their absorption. Mechanical digestion will play a role here, and this is where we physically break large chunks into smaller bits. And chemical digestion as well, where we break molecules apart further. Motility. This is movement by the muscular components of the digestive tube. It includes processes of mechanical digestion. So we'll look later at 2 of these processes called peristalsis and segmentation. Secretion. This is the release of digestive juices, and these digestive juices contain things such as enzymes and acids and bases. There's mucus. You've learned already a bit about bile and other products that facilitate digestion. Some digestive organs also secrete endocrine hormones, and we talked a little bit about this already, and these endocrine hormones help regulate the metabolism of nutrients and the overall digestive processes. Absorption. This is the movement of the digested nutrients through the gastrointestinal mucosa and into your internal environment, because remember that the digestive tract is really an extension of your external environment. Even though it's inside your body, it is considered part of the external environment, and material or nutrients do not become part of your internal environment until we actually absorb it. Okay, elimination. Obviously this is the process of getting rid of food and products that are not useful to our body, not needed. And then regulation. This is really just the coordination of digestive activity. So coordinating motility and secretion, for example, among other things. So the term digestion is the overall name that's oftentimes given to the processes of chemically and mechanically breaking down these complex food into simpler nutrients that can be easily absorbed and then utilized by our body. After food is ingested, so taken into the mouth, the process of digestion begins right away. So we'll speak now about the mechanical digestion of food. Mechanical digestion consists of all of the different various movements that we can call motility of the digestive tract. This process allows for the breaking of food into smaller pieces, so that's mechanical, and it also allows for the movement of food along the digestive tract or the GI tract. This, of course, will ultimately lead to elimination of feces from the body. Mechanical digestion begins in the mouth with the process of mastication, which is the chewing of food. This allows us to take in bulky food and chew it into smaller pieces, allowing us to then swallow the food in less of a bulky state. In addition, during mastication, food is mixed with saliva, which lubricates it, making it also easier to swallow. Now, in some cases, which we'll learn about later, the enzymes contained within saliva actually begin the process of chemical digestion. So in other words, with certain foods, both mechanical and chemical digestion begins in the mouth. Now, at this time when food enters your mouth, and you begin to mechanically break it down, it is called a bolus, which is spelled B-O-L-U-S. You should recall that deglutition is the technical term for swallowing, and it involves 3 main steps or stages: the oral stage, the pharyngeal stage, and the esophageal stage. And this is where your understanding of upper digestive tract anatomy will likely be helpful in understanding each of these stages. During the oral stage, food is going to move from the mouth into the oropharynx. So let's look at this top diagram here. If this is a sagittal view, if here's your oral cavity, here's your tongue, here's the bolus of food. The oropharynx is the part of the throat. Remember, the pharynx is the throat, the part of the throat that's behind or posterior to the oral cavity. So again, this is where food is moving from the mouth, so the bolus, into your oropharynx. This stage is voluntary, meaning that you have control over this act. During the oral stage, the bolus in green is going to be pressed up against the palate. You can see the palate up here. So remember, here's your hard palate, and here's part of your soft palate. Pressed up against the palate by your tongue, and then moved back into the oropharynx. And it's images like this that really show you how much muscle and how large your tongue really is. Stage 2 and 3 are both involuntary stages, meaning you don't have control over these acts, and they consist of the movement of food via contractions and gravity, as you'll see, from your throat or the pharynx into your esophagus, and then of course finally into your stomach. But let's look a little bit closer at the pharyngeal stage. To propel food from your pharynx into your esophagus, the pharyngeal stage, 3 openings must be blocked: the mouth, the nasopharynx, and the larynx. Let's look at each for a moment. The mouth. We have to close off the mouth so that food moves where we want it to go, which is away from the oral cavity. And so you can see the mouth closed off here. The nasopharynx. Well, if here's the nasopharynx, which is located just behind the nasal cavity, so this is the part of the throat behind the nasal cavity, we want this blocked off, again, to have food move in a downward direction, and the soft palate is going to act as a valve, really preventing food from entering the nasopharynx. So you can see that blockage happening here by the soft palate. In addition, and this is a little bit harder to visualize, but the larynx, which is this structure here, actually moves upward. And when it does that, here you can see a structure called the epiglottis. The epiglottis will then move throughout this process to cover the trachea so that food will move through the esophagus and not into the trachea. So really, we're closing off our mouth, we're closing off our nasal pharynx behind the nasal cavity, and we're closing off really our trachea when we swallow so that food moves in the proper direction into our esophagus. And then finally is the esophageal stage, and this is where food is moving along the esophagus downward toward the stomach. We said already that this is an involuntary stage and that both contractions and gravity, just like the pharyngeal stage, are going to work together to move food in this forward direction. Let's have a look now at the term peristalsis. And peristalsis is oftentimes described as being this wave-like ripple of the muscular layer of an organ. And it's not just specific to the esophageal, although we're talking about the esophagus on this slide. What happens is, if this is the esophagus, a bolus of food is going to stretch the GI wall. And when it stretches the GI wall, it triggers a reflex contraction of these circular muscles that make up the GI wall that push the bolus forward. So think of peristalsis then as really being a progressive forward kind of motility or movement along your GI tract. So the bolus causes a stretch. So you can see it's stretched out here compared to normal, which signals a contraction before it, and then that propels it forward. Then it moves forward, it stretches the GI wall wherever we are in this region, and then it signals a contraction before it, and so on and so forth. So you can see this progressive type of movement that's occurring. Okay, let's have a look at this right diagram here. This is showing you the pressure in the esophagus during swallowing. So after swallowing at the top here, here is your upper esophageal sphincter. the esophagus, and then your lower esophageal sphincter to the stomach. And this graph is showing you specifically the changes in, or the changing pressure measured in millimeters mercury inside the esophagus during the esophageal stage of deglutition that we talked about on the previous slide. And note that an area of high pressure moves progressively down the esophagus and eventually reaches the stomach. Note as well that during rest, which is interesting, throughout the esophagus, you can see that the smooth muscle during rest exhibits a slow rhythm of weak contractions and then an increase with actual swallowing of a bolus of food. Please remember, though, that peristalsis is happening all throughout the digestive tract to move food throughout. It's not specific to the esophagus, but on this particular slide, we were talking about the esophagus. Okay, so peristalsis, you now know, is a term that describes the forward propelling movement of food throughout our GI tract. And this is going to happen, this peristalsis, all throughout our GI tract. Another type of movement is called segmentation. And segmentation will be primarily talking about segmentation with respect to your intestinal tract. Segmentation here on the left of the slide can be described simply as a mixing movement. Segmentations occur when digestive reflexes will cause this forward and backward movement within a single region or segment of the GI tract. Now, this type of movement helps mechanically break down food particles, so you can see we're breaking down food particles into smaller parts. And also, this process is going to help mix food with digestive juices that are present in that area. Thirdly, segmentation will actually help put these small particles that we've broken down into closer contact with the digestive walls, or more specifically with the intestinal tract walls, so that we can absorb them. Okay, so it puts them in close contact then with the mucosal layer, where we will actually be able to absorb these nutrients once they're small enough. So when we talked about that reflexive action, when food moves through, we know that it's going to cause, or a bolus moves through the GI tract, we know that it causes peristalsis as well. But segmentation will also be stimulated, where you get these forward and backward contractions. Or better yet, I should say, these contractions that occur in front of the bolus and behind the bolus that encourage this segment and the food that's within this segment to be mechanically broken down and chemically digested. In this second diagram, we're looking more specifically at the stomach. Mixing actions in the stomach include propulsion, which is a forward movement, and retropulsion, which is a backward movement. So you can see those terms listed here and the corresponding arrows. So forward movement and backward movement. Food mixing with gastric juice in the stomach now takes on a different name. And that name, from where we were as a bolus to now mixed with digestive juices, is more specifically now called chyme. Okay, so food that's mixed with digestive juices takes on the name chyme. Now, as contractions within the stomach get stronger, what will end up happening is some of this liquid chyme is going to be ejected. This happens about every 20 seconds, where it's ejected into the first part of the duodenum by bypassing the pyloric sphincter, which is right here. And food will be sent then in the form of chyme into your duodenum of the small intestine. The process of emptying the stomach, or gastric emptying, takes about 2 to 6 hours after a meal is consumed, and that is dependent on the person, whether you fall between 2 and 6 hours, sometimes even dependent on the time of day, but also dependent on the type of food that is within your stomach. Because the volume in the stomach is large and that of the duodenum is small, gastric emptying must be regulated. Otherwise, you will really overburden the duodenum if you send everything from the stomach into the duodenum. So we control it primarily by two mechanisms. One is hormonal and one is nervous. So hormonal being related to the endocrine system, and nervous will sometimes be referred to as neural control. Fats and other nutrients in the duodenum. So when fats and nutrients are present here, this will stimulate the intestinal mucosa to release a hormone called gastric inhibitory peptide, so GIP. Gastric inhibitory peptide will be then released into the bloodstream. Then through the bloodstream, it will reach the stomach wall. And what this hormone does is have an inhibitory effect, as the name suggests, on gastric muscle, decreasing its peristalsis, or that forward movement, and this will then slow the passage of food or chyme into the duodenum. So once again, food present here will stimulate a hormone that will be released into the circulation, reach the stomach, pretty much telling the stomach to slow down the contractions, which will ultimately reduce the quantity of food or chyme that leaves the stomach and enters the duodenum to prevent overburdening the duodenum with food or chyme. Nervous control results from receptors that are found within your duodenal mucosa that are very sensitive to the presence of both acid, so pH changes, and distension. So the distension then is related to the mechanoreceptors, and pH related to the acidity. So they're sensitive to these variables. All right, so what will happen next then is when acid is present and when distension occurs, the duodenal mucosa picks it up, and sensory and motor fibers that are found in your vagus nerve, so this process is called a vagal reflex, will cause this reflexive inhibition of gastric peristalsis. This is also known as the enterogastric reflex, where, once again, when the duodenum stretches and when there is acidic contents within it, then it will stimulate the vagus nerve, which will end up inhibiting gastric peristalsis via what's called the enterogastric reflex. So a quick refresher here on motility. We talked about peristalsis being that progressive forward movement of food throughout your GI tract, and that it occurs in your esophagus, your stomach, it occurs in your intestines, but segmentation was more specific to your intestines, where contractions will occur that will take food where it is in a segment and mechanically break it down, encourage it to mix with digestive juices in the area, but also when food is small enough, put it into contact, close contact with the intestinal mucosa in order for us to actually absorb those nutrients. So intestinal motility then includes both peristalsis or peristaltic contractions and segmentation. After leaving the stomach, chyme takes about 5 hours to pass all the way through the small intestine. Peristalsis rate increases until near the end of the jejunum. So the order of the small intestine from the start to the end is the duodenum, jejunum, and ileum, or DJI. And there are several mechanisms that are involved in the control of this intestinal motility. Peristalsis is regulated in part by just intrinsic stretch reflexes that occur. And we really already talked about those stretch reflexes in this image. It is also thought to be stimulated, this peristalsis, by a hormone called cholecystokinin, or CCK, which is secreted by endocrine cells, because it is a hormone, endocrine cells of the intestinal mucosa when chyme is present. So when chyme is present in the intestinal region, the mucosa will release, the endocrine cells of the intestinal mucosa will release a hormone called CCK, which controls the rate of peristalsis throughout the intestines. Segmentation in the duodenum and the upper part of the jejunum will mix the incoming chyme with digestive juices from the pancreas, from the liver, from the intestinal mucosa, as discussed in previous lectures. And of course, we also know that the segmentation is going to put food, nutrients in close contact with the intestinal mucosa for absorption when they are ready to be absorbed. But before they're ready to be absorbed, they need to be broken down and mixed with these digestive juices. So we can say then that segmentation really augments or contributes to chemical digestion by mixing the food with digestive enzymes. And what digestive enzymes? Well, we're going to be talking about this, but speaking very generally, they're mixing with these enzymes that act as extracellular, so they're outside cells, these extracellular organic catalysts, which ultimately break down food. But we'll come back to that. Mixing will also promote hydrolysis, which is a chemical process where you have a compound that unites with water and then splits it into simpler compounds, so ultimately just further breaking down the food. When I speak about digestive enzymes, I'm speaking of chemical digestion versus mechanical digestion, which was already discussed. So we're focusing now on chemical. Most digestive enzymes are synthesized and secreted as inactive proenzymes. And so I'll highlight in the diagram what this inactive proenzyme looks like. So these are released, then produced by certain organs, and then released in the form of an inactive proenzyme. Now, enzymes that break apart proenzymes and really then converting them to active enzymes are called kinases. So kinases then break down proenzymes, which are inactive, which are digestive enzymes. It'll be the kinases that activate them. Digestive enzymes, like other enzymes in our body, are very specific in their action. They have a specific role. Most enzymes catalyze a chemical reaction in both directions, as you might remember from basic chemistry. But enzymes need an optimal environment in order to function properly. And so we're going to talk about that optimal environment for a moment in this bottom diagram. Digestive enzymes function optimally at a specific pH and become inactive if the pH deviates beyond these very narrow limits. Different enzymes function optimally, though, at different pH levels. Pepsin, for example, versus trypsin. Pepsin operates within a low pH range, whereas trypsin operates within a higher pH range. Both are protein digesting enzymes. Pepsin is a gastric secretion and trypsin is a pancreatic secretion, as you'll see soon. But I wanted to go back to this diagram here to remind you of proenzymes and kinases, but to show you how they actually function. A proenzyme, once again, is a digestive enzyme that's inactive, that's released from some accessory organ or some organ, digestive organ that releases it. It is combined with this prosthetic group in pink. When kinases are present, which we know are involved in actually activating the proenzyme, it will bind to it. When it binds to it, it causes that prosthetic group to leave along with the kinase. And this form now is an active proenzyme. This active proenzyme or digestive enzyme is now able to bind with some sort of complex nutrient that your body consumed and break it down into its smaller, simpler products. Over the next handful of slides, we're going to be talking about the digestion of carbohydrates, proteins, and fats. Now, in addition to the ingestion of these chemical substances, we also ingest vitamins, minerals, salts, and water. So these here are the 6 main types of chemical substances that we consume. However, it's the first 3, carbs, protein, and fats, that need to be chemically digested in order for us to absorb it. Carbohydrates are saccharide compounds, so you can see the term saccharide here. This means that their molecule will contain 1 or more saccharide groups. And as you can see, it's either going to be mono, di, or poly. Polysaccharides, notably, examples would include starches that we consume, and glycogen, which is a stored form of glucose, contain many of these saccharide groups, hence the name polysaccharide. Whereas disaccharides contain 2 of these groups, and mono, being 1, contain 1 of these groups, or contain 1 group. Polysaccharides, to begin here, are hydrolyzed or broken down into disaccharides, because we need to break down these larger substances into smaller substances in order for our body to actually absorb it. So we break these larger substances as polysaccharides from, let's say, the starches that we eat into smaller forms, so a disaccharide, and this is done by enzymes known as amylases. So amylases are produced by and found in your saliva and your pancreas. And so they are called salivary amylase and pancreatic amylase based on where they are initially produced. So these enzymes then will break down these polysaccharides, so let's say starches, into a smaller form such as disaccharides. Another important point to make has to do with other properties of saliva. Saliva contains sodium bicarbonate, and sodium bicarbonate will act to increase pH or reduce acidity. And this contributes to overall good oral health. Now, disaccharides have to be broken down into monosaccharides, and the specific enzymes that catalyze this reaction are sucrase, lactase, and maltase. When you see the suffix ase, we're referring to enzymes. And so it might make sense to you then that sucrase is the enzyme that breaks down sucrose. Lactase will break down lactose, OSE as a suffix refers to sugars, and maltase will break down maltose. These enzymes are actually called brush border enzymes, as indicated on this image, because they're located in the cell membrane of epithelial cells within your digestive tract, and their presence then, of course, will help break down disaccharides into monosaccharides. So let's put this together in the image. When we consume polysaccharides, they are going to be broken down in our body by salivary amylase within our saliva and pancreatic amylase from our pancreas, which will break down polysaccharides then into smaller forms called disaccharides. Disaccharides can be then further broken down into their smallest form as a monosaccharide, which is something we can actually absorb in our body, and this is done by brush border enzymes sucrase, lactase, and maltase, which will break down their corresponding sugar into smaller forms. Now let's talk about protein digestion. Protein compounds have very large molecules made up of folded or twisted chains of amino acids, often hundreds in number. And the goal when we consume protein is to be able to break it down into its smaller form, smallest form being amino acids, which our body can actually use or rather absorb and use to produce, for example, many different proteins within our body. Enzymes called proteases will catalyze the hydrolysis of protein first into a variety of intermediate compounds called proteoses and peptides, which are really just shorter chains of amino acids. But this starts the breakdown process. These proteases exist in both gastric and pancreatic juice. The gastric juice protease is called pepsin, and the pancreatic juice protease is called trypsin. So pepsin and trypsin then are proteases that will break down protein into peptides and proteoses. Peptides are broken down into their smallest form as amino acids by intestinal peptidases. In other words, these are peptidases or enzymes that are found in the intestines. And proteoses are broken down by proteases that are produced by or found in pancreatic juice. Both use hydrolysis. So you can see this term, this metabolic reaction being written many times so far. And the final product ends up being the amino acids that we use as a building block for many different functions in our body. So we can say that each kind of protease within our body is going to be catalyzing the breaking apart of a specific peptide bond. Now let's look at fat digestion. Because fats are insoluble in water, think of how oil and fat don't mix, they must be emulsified first. In other words, they must be dispersed into smaller droplets, and this has to be done mechanically. This has to happen before we can actually digest it and break it down. The emulsifying agent is called bile, which, if you had a chance to watch the last online lecture, is made in the liver and stored in the gallbladder. First, bile emulsifies the fat within the small intestine. So once fat reaches the small intestine, then bile will emulsify it, and this allows for enzymes to actually access it. So think of bile as like a detergent. Bile sort of acts like a soap that helps break down the fat just enough to allow for the enzymes to access it. So if we have a look here at this large fat drop that comes in, what bile will do, produced in the liver, stored in the gallbladder, and released into the small intestine when fat is present, will surround different components of the fat, forming smaller fat droplets. It just makes it easier for enzymes, in this case pancreatic lipase, to actually access and really break down the fat. Pancreatic lipase, lip means, or lip here refers to fat. Think of lipids, and ACE is an enzyme. This is your main fat digesting enzyme produced by and secreted by the pancreas into your small intestine, and it contributes to that chemical digestion or hydrolysis of fat. The dietary fat that we consume is usually in the form of triglycerides, and so what we'll be doing then through this process of breaking down fat is breaking the triglycerides down into their smallest components, fatty acids, monoglycerides, and glycerol, which you can see were done by hydrolysis by your pancreatic lipase. Now, before we move on, I want to mention what a micelle is. You can see a micelle in the image, and it's really just a small aggregate of your mixed lipids and bile acids that are suspended within your intestines. So you can see 4 micelles here. Now, once we break down fat into its smaller form, fatty acids, monoglycerides, and glycerol, it has to be absorbed into our body. But what we'll see on an upcoming slide is that proteins, when we break them down into amino acids, and carbohydrates, when we break them down into monosaccharides, they will be absorbed into our bloodstream. But fats are different. They actually get transported, instead of immediately in our blood, which is primarily water, they get transported in lacteals, as you'll see shortly. These are just lymphatic vessels. The final point on this slide is that certain components of food resist digestion, and so they're eliminated from the intestine in the form of feces, and this is called the residues of digestion. If we lack enzymes to digest something, then it will pass through undigested. Cellulose is a great example. It's a type of dietary fiber, but there are other substances as well that make up our feces that resist digestion. Gastric juice is secreted by gastric glands, and so we call these exocrine glands, which have ducts that lead to the gastric lumen by way of gastric pits. Now that's a statement that we have discussed already in a previous lecture. Gastric juice contains mucus and water in order to lubricate, protect, and to facilitate mixing of the chyme. It also contains digestive products such as pepsin. Pepsin is the active form of this enzyme, but before it's active, it's called pepsinogen. So pepsinogen is basically an inactive form of pepsin. So that's an important point for you to know. Pepsinogen is secreted by chief cells, which have been introduced to you already. Chief cells are found within gastric glands in the stomach. Pepsin, and this is a very important point, is the protease or the enzyme that begins the process of protein digestion, which we mentioned earlier. But first, pepsinogen, which is in its inactive form, needs to be converted to its active form, which is pepsin. And the question is, how is this done? How do we take this pepsinogen that was produced by chief cells to become its active form of pepsin, where it can begin the process of protein digestion? Well, this is done by hydrochloric acid, or HCl. Hydrochloric acid then converts pepsinogen to pepsin, and it does this by decreasing the pH of chyme. Well, let's make a little bit more sense of this. First of all, recall from a previous lecture that hydrochloric acid is produced and secreted by parietal cells found within the gastric mucosa. Up here we have the gastric lumen, so this is the opening of the stomach where your food is. Here are your gastric cells, and then here is your blood capillary. Within our gastric lumen, pepsinogen will be present in this area. When it's secreted or produced and secreted, it will be found within the gastric lumen. It's inactive, however. In order to activate it, what we need to do is change the pH of the gastric contents. And how this is done is with this hydrogen potassium pump that will pump hydrogen, which we know contributes to acidity, into the gastric lumen by way of removing potassium. In addition, chloride will be pumped into the gastric lumen or will move through this channel into the gastric lumen, where hydrogen will combine with chloride to form hydrochloric acid. This will make the gastric contents more acidic, which will then activate pepsinogen into its active form, which is pepsin. And so this goes back to what we discussed previously about how enzymes need to be in a certain pH in order to be in their active form. Of interest note only, if you've ever known somebody who produces too much acid within their stomach and maybe they're prone to ulcers, so stomach acidity needs to be reduced, doctors can prescribe proton pump inhibitors, which really inhibit this pump here, which will reduce the overall stomach acidity. Okay, let's now have a look at the control of these gastric secretions. Stimulation of gastric juice secretion occurs in 3 phases that are controlled by reflexes and chemical mechanisms. Because stimuli that activate these mechanisms arise in your head, your stomach, and your intestines, they're therefore called your cephalic, referring to head, gastric, and intestinal phases, respectively. The cephalic phase is also spoken of as the psychic phase because it's mental factors that activate this mechanism. For example, the sight or the smell or the taste or even the thought of food that is pleasing to an individual will activate control centers that are found within your medulla oblongata of the brainstem. You can see the activation here from something that you enjoy if you like ice cream, which will send out parasympathetic stimulation via your vagus nerve to gastric glands, and this directly stimulates gastric juice secretion and also stimulates the release of gastrin, which prolongs and enhances this effect. So we spoke about gastrin during one of our previous lectures. During the gastric phase, the presence of food in your stomach, specifically when the presence of food causes distension, triggers local and parasympathetic reflexes to occur. So you can see the distension in this image. This will end up causing an increase in gastric juice secretion as well as gastrin in order to further enhance this effect or amplify the release of gastric juices. Now, as food moves from your stomach into your duodenum, the presence of fats, carbohydrates, and acid will stimulate hormonal and nervous reflexes, as mentioned earlier, that inhibit stomach activity. This likely involves hormones such as GIP that we talked about. Also, there's another hormone called secretin that will come up shortly, and cholecystokinin or CCK. This stage is certainly less well understood than the other two, but it is thought, again, that gastric secretions are inhibited when chyme that contains fat, carbohydrates, and acid is present within the duodenum, which will slow down the secretion into the duodenum in order to not overburden the duodenum. Pancreatic juice is secreted by exocrine acinar cells of the pancreas. Although pancreatic juice is mainly water, pancreatic juice also contains various digestive enzymes. All of these enzymes are secreted as inactive proenzymes, as mentioned earlier. The acinar cells of the pancreas produce proteases, lipases, nucleases, amylases, and sodium bicarbonate in order to increase pH. Much of these we've already spoke about. Several hormones are released by the intestinal mucosa and are known to stimulate pancreatic secretions. Secretin and CCK are examples. Secretin is going to evoke the production of pancreatic fluid that's low in enzymes but high in bicarbonate, and this acts to neutralize the chyme. So, the presence of acid in the duodenum will stimulate the release of secretin in order to neutralize the chyme. CCK, or cholecystokinin, is secreted by intestinal mucosa in the presence of fats and other substances, and this will then cause the pancreas to increase exocrine secretions that are higher in enzymes. It will inhibit hydrochloric acid. It will stimulate contractions of the gallbladder in order to release bile. Now we're going to move on to talking about bile a little bit more detail. Bile, you know, is important in fat digestion and that it's synthesized in the liver and stored and really concentrated in the gallbladder. But what is bile made up of? Bile contains several substances that aid in digestion, specifically lecithin and bile salts, which are going to emulsify the fat by encasing them into these shells to form tiny micelles, as we looked at on a previous slide. And what that does is it makes them water soluble. It makes them easier to digest. Bile also contains small amounts of sodium bicarbonate, which helps to neutralize the chyme in the small intestine, and we know that a specific pH is necessary for optimum enzyme function. Bile also contains several substances that are ultimately designed for elimination, such as cholesterol, products of detoxification in the body, and bile pigments. These are waste products that the liver excretes with the bile in order to get it out of our body. And so it will ultimately be eliminated then in your feces. We already know that bile is continually produced and secreted by the liver, but two hormones called CCK, or cholecystokinin, and secretin are involved in stimulating the ejection of bile from the gallbladder, where the bile is stored until it is needed. So you can see that secretin and CCK play multiple different roles. We just spoke about their role with pancreatic secretions on the previous slide. And now we discussed about its role with bile ejection from the gallbladder. Absorption primarily occurs in the intestine, meaning that your stomach absorbs very little. Your intestine, and you can see it here, alcohol and aspirin, but not even alcohol in its entirety. But most of the absorption very clearly occurs within the small intestine, with even very little overall occurring within your colon and none at all within the rectum. Now, what contributes to absorption? Well, it's the presence of intestinal villi and microvilli that increase the absorptive surface area of the intestinal mucosa. Most absorbed substances, I already mentioned, will enter the blood stream, with the exception of fat that enters the lymphatic system by way of those lacteals. So let's have a look at some of these terms that I mentioned. Within our intestine, we have these projections that contribute to an increase in surface area called villi, intestinal villi. Then on top of that, and it's difficult to see in this image, but there are additional structures that contribute to an increase in surface area called microvilli, just smaller villi. So food will be absorbed all throughout here. All throughout each villi and microvilli within the small intestine. But you can see that within each villi, or within one villus, what we have is blood vessels. And this is where we're going to be absorbing most nutrients, is into this venous system here. And then substances such as fat are going to be entering the lacteal, or the lymph lacteal that's found within the villi. Okay, mechanisms of absorption. We're going to talk about how sodium, glucose, and amino acids, when present within the gastrointestinal lumen, or the lumen of our intestinal tract, how does it get into our bloodstream? But before that, you should know that particles can actually move from the lumen of our intestinal tract into our capillary system by way of paracellular or transcellular absorption. We're going to be focusing on transcellular, but the difference between the two is as follows. Paracellular means that substances or particles can move in between cells. So if this is a cell, this is a cell, this is a cell, in between cells is paracellular absorption. Whereas transcellular absorption means that particles, or nutrients in this case, are going to be moving through the cell. That's transcellular absorption. So that will be our focus. Mineral ions, such as sodium, when we consume them and they end up in our intestinal lumen, can be absorbed into our cell. Then from there, with the use of ATP, can be brought into our blood capillary. Water will move via osmosis, but nutrients such as amino acids and glucose will move via active co-transport with the mineral ion sodium. So let's see how that looks for each. Up at the top here we have glucose, represented by G, and then of course sodium, Na+. When glucose and sodium are both within the intestinal tract, they can be absorbed together, so this is the co-transport, absorbed together into the cell. Then from there, sodium, as we know, will end up going into the blood vessel with the use of ATP, whereas glucose can enter the blood capillary very passively. The thing with glucose is that it's a large molecule, and it's also water soluble, and so it can't pass through this region here, the brush border of the intestinal mucosa. And so its size and the fact that it doesn't like lipids, because it's a water soluble molecule, and we know that plasma membranes are lipid, it can't be absorbed on its own. That's why it needs to be co-transported. And amino acids are very similar in that fashion, that they will be co-transported for similar reasons into the cell, and then sodium leaves through an active transport, active because it's using ATP, and then amino acids can move passively into the blood capillary. So where are the nutrients such as amino acid and glucose and the blood that's coming from the digestive tract, where is it going? Let's go back to this slide here just to show you that once we've absorbed what we've just spoken about, where is this going? Where does it travel to? Does it go right to all of our organs to give them the nutrients? No. First, it actually goes to the liver through what's called the hepatic portal system. And this is important because our liver will then detoxify this blood that's come from our digestive tract before it sends it to the heart, which would then, of course, go through its pumping action through our pulmonary circuit and systemic circuit to provide our body with oxygen and nutrient-rich blood. On the previous slide, we spoke about how proteins, once broken down into amino acids, get brought into the blood from our digestive system, and how carbohydrates, once they're broken down into monosaccharides, such as glucose, get brought into our bloodstream from the digestive tract. But what about fats or lipids? We already know that lipids don't get brought into the blood vessels, that instead they are brought into what are called lacteals, which are an intestinal lymph vessel, oftentimes represented by the color green. Fatty acids, monoglycerides, and cholesterol are all lipids, and they are transported with the aid of bile salts into the lumen, from the lumen of the intestinal tract to absorbing cells of the villi. So what does that mean? Well, let's have a look at the lumen of our intestinal tract and how we have this fat that's present here. We have bile, which is going to emulsify the fat and form these droplets, these smaller droplets, which will allow for enzymes like pancreatic lipase to really get to this fat and break it down further, allowing for these individual structures, individual lipids, to pass just by simple diffusion through the plasma membrane, through this brush border here. Now, why can it pass right through? Well, it's a fat, and so is the plasma membrane, so it can pass easily right through into the cell. So these are the epithelial cells then of your intestinal tract. Okay, now these fatty acids and monoglycerides that have entered into the epithelial cells of the gastrointestinal tract need to end up within the lymph lacteal, and what happens is the formation then, the sort of repackaging into what's called a chylomicron. And a chylomicron is really this water-soluble molecule that can travel into the lacteals where it will be transported throughout the body. And then these lymph lacteal vessels will end up joining in with the rest of the lymphatic system. The process of elimination is simply the expulsion of the residues of digestion or feces from the digestive tract. Formation and storing of feces is the primary function of your colon. The act of expelling feces is called defecation. Defecation is a reflex that's brought on by the stimulation of receptors in the rectal mucosa. Normally the rectum is empty. It's empty until there is this mass peristalsis that occurs, which moves fecal matter out of the colon and into the rectum. And when this happens, the rectum distends and it will produce the desire to defecate. This involves the relaxation of different sphincters that are found in this region as part of the anal region. So this process then brings upon defecation. Constipation. This occurs when contents of your distal part of the colon and rectum move at a rate that is slower than normal. And so what is normal can range from 3 times a day, which is actually considered to be ideal, to 3 times a week. Now, this depends on a variety of factors such as the amount, type, and rate of food that's eaten. When motility in the distal colon and the rectum slows significantly, then it permits water being taken from the fecal mass, so pulling water from the fecal mass into your intestinal tract. And then if your stool has less water, it becomes harder, and this is then constipation. Now, as far as frequency of bowel movements, if somebody is eating 3 normal meals a day, then they ideally should be defecating 3 times a day, but this is not seen as frequent, especially in this day and age with a lot of the processed foods that are eaten and with sedentary lifestyles. Now, diarrhea can occur as the result of increased motility of either the SI, so the small intestine, or the large intestine. When chyme moves through the, for example, small intestine too quickly, it reduces the amount of absorption of water and electrolytes. We know that most absorption occurs within the small intestine, so if we don't leave it there, if it doesn't stay there long enough for us to actually absorb the water and electrolytes from it, then we end up losing it as part of our stool. Diarrhea can also result from bacterial toxins that damage the water reabsorption mechanisms of the intestinal mucosa. Also, if you have a bacterial infection in your GI tract that causes diarrhea, the bacteria can be powerful enough that it can actually draw water from your intestinal cells, creating excessive watery diarrhea. So this large volume of material, including excess water that arrives in the large intestine, really exceeds the large intestine's ability to absorb it, and so a watery stool results. Prolonged diarrhea can obviously be very serious and even fatal, especially in infants, because of loss of water and electrolytes, and in the case of an infant, their reserve of water and electrolytes is going to be more limited than that of an adult. Okay, on our final slide, this is just showing you digestion and its relationship to the other systems within our body. So in order for digestion to occur, it needs other systems to contribute to it. So we talked about the role of the nervous system and the endocrine system in regulating digestion. Of course, oxygen is necessary for any tissue to function, and so our respiratory systems and circulatory systems need to be functioning optimally for any cell and the tissue that it makes up to survive. And of course, many of the hormones that are produced end up needing to be need to enter our circulatory system to then travel to our digestive organs to have their effect. Our integumentary system, primarily our skin and our skeletal system, acts as a supportive and protective function for our digestive organs, and our muscular system is needed for us to ingest food, chew food, swallow food, and then what's not put in here is just the peristalsis and segmentation that we spoke about earlier, and then defecation. And some of these processes are voluntary, whereas others are involuntary. And that's where when you talk about the muscles that make up our intestinal tract and our stomach and our esophagus, they're made of smooth muscle, and smooth muscle is a muscle type that we do not have voluntary control over. That's the end of today's lecture. Thank you so much for listening.